So for this video, we're going to be looking at the chromosomal basis of inheritance. And I'm going to make this very short and sweet because I find that this part of the PowerPoint and the book is very wordy. Basically, this section is included to show that Mendel's laws also follow chromosomal theory of inheritance, which means that genes occupy specific loci or positions on the chromosomes, and that chromosomes will undergo segregation and independent assortment during meiosis, which is what Mendel's laws show, that the law of segregation depends on separation of homologous chromosomes in anaphase 1, and the law of independent assortment depends on alternative or different orientations of chromosomes in metaphase 1. So this is just to show you that if you was to follow the chromosomes throughout meiosis, you would still end up with the same chromosomal probabilities as you would in a dihybrid cross. And this is just to show you that if, once again, mixed egg and sperm, the chromosomes still have a specific probability of separating independently, which is the same as what can be found here in a dihybrid cross. So there was an irregularity, I guess, something that they found, Bateson and Punnett, when they were studying plants, found that they didn't always get the 9:3:3:1 ratio in the second generation. And so this is what they found to be known as linked genes, in that there are some genes that are so close together on the chromosome that they tend to be inherited together. And this can be seen here in the experiment with purple flowers and their pollen grains, if they have long pollen or round pollen. And so you can see the prediction of the number of offspring that should have occurred with these phenotypes, but what you'll notice here is that actually many more of them were actually purple and long compared to what should be predicted, and less were purple and round and red and long compared to the prediction. So the observed numbers are not the same as what was predicted. And so how do you explain this? It has to do with genes being located very close together on the chromosomes. So this is just showing you the chromosomes here, and then if you have most gametes either end up being having either both dominant or both recessive alleles, and then if you make those sperm and eggs, you'll get individuals that have a more 3-to-1 output. So purple and long being the dominant phenotype compared to the one red and round. And so this would actually almost eliminate the purple and round and the red and round traits being put together, because these traits are so close together on the chromosomes, they're inherited together. And so linked alleles can be separated during crossing over. So they're not always stuck together, but they can be separated during crossing over, which is not as common as a normal gamete. So the percentage of recombinants, or this new recombination of alleles, is called the recombination frequency. And so this is just showing you how you can have a tetrad during meiosis, and then crossing over occurs in prophase 1. And so you can get either the same parental gametes, or you can get new recombinant gametes that are different from the parental gametes. And so you can see this, you can actually do an experiment of this with flies, where you can have a gray body with long wings, a black body with vestigial wings, which means they don't act as wings anymore, or they're not supportive. And you can make these individuals, and you can get these resulting phenotypes from the offspring, where you can get parental phenotypes, those seen in the parents, but then you can get some new ones from recombinant phenotypes. And so if you just do the math, the numbers on here, which I'm not requiring you guys to do any math, but the recombination frequency is around 17%. And so this is just explaining the genetics behind that, which I'm not too worried about right now. But it is this recombination frequencies that scientists have used to actually identify specific genes on chromosomes, because if you can figure out the frequencies, you can figure out how closely linked they are, how close together they are. And so from that information and many different studies, for example, they have typed out one of the chromosomes of these flies here. And they do the same thing with human genes, other species, but this way they are able to locate and identify different genes and traits on a chromosome.